Metabolism of microorganisms. What is metabolism? Metabolism refers to all of the biochemical reactions that take place in an organism. These processes are divided into two general categories: catabolism and anabolism. Anabolism builds larger organic compounds, such as carbohydrates and proteins, from simpler monomers, including glucose and amino acids. Catabolism hydrolyzes these polymers into simpler molecules such as carbon dioxide, ammonia, and water. From an energy perspective, anabolic reactions form bonds, which requires energy. Such energy requiring processes are considered endergonic reactions. In contrast, catabolic reactions break bonds, releasing energy. These processes are called exergonic reactions. Enzymes catalyze all chemical reactions in cells. Enzymes are usually proteins. Enzymes increase the probability of a chemical reaction taking place, while they themselves remain unchanged. Enzymes are able to accomplish in fractions of a second what otherwise might take hours, days, or even longer to happen spontaneously under normal biological conditions. For example, even though organic molecules, like amino acids, have functional groups, it is highly unlikely that they would randomly bump into one another in the precise way needed for the chemical reactions to take place. Thus, the reaction rate would be very slow if it were not for the activity of these enzymes. There are four characteristics that are common to all enzymes. First, enzymes are reusable. Second, enzymes are highly specific. Third, enzymes have an active site. And fourth, enzymes are required in minute amounts. First, we will look at how enzymes are reusable. Once a chemical reaction has occurred, the enzyme is released to participate in another identical reaction. In fact, the same enzyme can catalyze the same reaction 100 to 1,000 times every second. Enzymes are also highly specific. An enzyme that functions in one type of chemical reaction will usually not participate in another type of reaction. This means there must be thousands of different enzymes to catalyze the thousands of different chemical reactions used in metabolism. Enzymes have an active site. Each enzyme has a special pocket or cleft called an active site, which has a specific three-dimensional shape that is complementary to a specific reactant or substrate. The active site positions the substrate in such a way that it is highly likely for a chemical reaction to occur. Enzymes are required in only small amounts. Because an enzyme can be used thousands of times to catalyze the same reaction, only minute amounts of a particular enzyme are needed to ensure that a fast and efficient metabolic effect occurs. Many enzymes can be identified by their names. Most enzymes have a name that ends with ase. For example, nucrase is the enzyme that breaks down sucrose. Ribonuclease digests ribonucleic acid. In terms of anabolic metabolism, polymerases link together nucleotides, and transferases link together the NAG and NAN units to build the bacterial cell wall of peptidoglycan. Let's explore the mechanism of action of an enzyme. First, an enzyme molecule has an active site where it binds the substrate molecule. The substrate will then bind to the active site to form an enzyme-substrate complex. Then, the enzyme breaks apart the substrate molecule, and two end products result from such reaction. The enzyme molecule remains unchanged and is recycled. Although this example shows an enzyme hydrolyzing a substrate, enzymes also catalyze dehydration reactions, which would bind two or more molecules together, forming a larger molecule. Enzymes function by aligning substrate molecules in such a way that a reaction is highly favorable. In hydrolysis reactions, the three-dimensional shape of the enzyme's active site recognizes and holds the substrate in an enzyme-substrate complex. While in the complex, chemical bonds in the substrate are stretched or weakened by the enzyme, causing the bond to break. In a synthesis reaction, by contrast, the electron shells of the substrates in the enzyme-substrate complex are forced to overlap in the spot where a chemical bond will form. Thus, recognition of the substrate or substrates is a precisely controlled, non-random event. Enzymes play a key role in metabolism because they provide an alternate reaction pathway of less resistance, that is, with a lower activation energy barrier. They assist in the destabilization of bonds and the formation of new ones by separating or joining atoms in a carefully orchestrated fashion. Some enzymes are made up entirely of protein. Other enzymes, however, contain small non-protein substances called cofactors. When a non-protein cofactor is a small organic molecule, we refer to it as a coenzyme. Cofactors and coenzymes will participate in the catalytic reaction. Enzymes often team up in metabolic pathways. Metabolism often involves a series of chemical reactions controlled by separate enzymes. A metabolic pathway is a sequence of chemical reactions, each reaction catalyzed by a different enzyme, in which the product of one reaction serves as the substrate for the next reaction. The pathway starts with the initial substrate and finishes with the final end product. The products of the in-between stages are referred to as intermediates. Metabolism can control and be controlled by enzymes. Enzyme activity can be inhibited. Let's look at the metabolic pathways and enzyme inhibition. In a metabolic pathway, a series of enzymes transforms an initial substrate into a final end product. If excess final end product accumulates, it will feed back on the first enzyme in the pathway and inhibit that enzyme's function by binding to another site on that enzyme. In competitive inhibition, the inhibitor will have a structure that is similar to the normal substrate and will compete for the enzyme's active site. Competitive inhibition would reduce the productivity of the metabolic pathway by slowing down or stopping the pathway altogether. Enzymes are sensitive to changes in temperature, pH, and certain chemicals. High temperature can denature a protein, perhaps bringing the metabolism to a sudden halt. An increase or decrease in protons affects the pH and will interfere with an enzyme's reaction rate. Extreme changes in pH can lead to not only enzyme denaturation, but also can lead to enzyme and metabolic inhibition. There are also certain chemicals that can inhibit enzyme action. Alcohols and phenols inactivate enzymes and precipitate proteins, making these chemical agents effective antiseptics or disinfectants. Other natural chemicals interfere with enzyme action, such as antibiotics like penicillin. Enzyme inhibition through pathway modulation. The same chemical reaction does not occur in a cell at all times, even if the substrate is present. Rather, cells regulate the enzymes so that they are present or active only at the appropriate times needed during metabolism. One of the most common ways of modulating enzyme activity is for the final end product of metabolic pathway to inhibit an enzyme in that same pathway. This is feedback inhibition. If the first enzyme in the pathway is inhibited, then no product is available as input for the rest of the pathway. Such feedback inhibition is typical of many metabolic pathways in cells. Non-competitive inhibition. In general, when the final end product or end molecule binds to an allosteric site, which is separate from the active site on the enzyme, the shape of the active site changes and can no longer bind with the substrate. This type of modulation is referred to as non-competitive inhibition. Competitive inhibition. Another way of modulating an enzyme is by blocking its active site so that the normal substrate cannot bind. Such competitive inhibition occurs in the following way. If a molecule resembles the normal substrate, it can bind reversibly to the active site, competing with the normal substrate. Energy in the form of ATP is required for metabolism. ATP is the universal energy currency in all cells. In many metabolic reactions, energy is needed along with enzymes for reactions to occur. The cellular energy currency is a compound called ATP, which stands for adenosine triphosphate. In bacteria and archaeal cells, the ATP is formed on the cell membrane, while in eukaryotic cells, these reactions occur primarily in the mitochondria. An ATP molecule acts like a portable battery. It provides the needed energy for activities such as binary fission, flagellar motion, active transport, and spore formation. On a more chemical level, it fuels protein synthesis and carbohydrate breakdown. It is safe to say that the major share of microbial functions depend on a continual supply of ATP. Should the supply of ATP be cut off, the cell dies very quickly, as ATP cannot be stored. ATP molecules are relatively unstable. The three phosphate groups all have negative charges on them. Like charges repel, so these phosphate groups and ATP being tightly packed together makes for a very unstable molecule. Breaking the so-called high-energy bond that holds the last phosphate group on the molecule produces a more stable molecule called adenosine diphosphate, or ADP. Breaking the high-energy bond releases energy. The hydrolysis of unstable phosphate groups in ATP molecules to a more stable condition is what drives other energy-requiring reactions through the transfer of phosphate groups. The addition of a phosphate group to another molecule is called phosphorylation. Because ATP molecules are unstable, they cannot be stored. Therefore, microbial cells synthesize large organic compounds like glycogen or lipids for energy storage. The chemical energy in these molecules can be released in catabolic reactions and used to reform ATP from ADP and a phosphate group. This ATP-ADP cycle occurs continuously in cells. It has been estimated that a typical bacterial cell must reform about 3 million ATP molecules per second from ADP and phosphate to supply its energy needs. The ATP molecule is composed of adenine and ribose, bonded to one another and bonded to 3 phosphate groups. When the ATP molecule breaks down, it releases the last phosphate group and energy is released. At this point, the ATP has become ADP, adenosine diphosphate. The freed phosphate group can activate other chemical reactions through phosphorylation. For the synthesis of ATP, energy and a phosphate group must be supplied to an ADP molecule. Metabolic pathways are coupled to ATP and ADP cycling. Glucose contains stored energy that can be extracted. Glucose is a primary source for generating ATP. Virtually all cells make ATP by harvesting energy from exergonic metabolic pathways, such as the hydrolysis of food molecules. Such a process is called cellular respiration. If cells consume oxygen in making ATP, the process is called aerobic respiration. In other instances, cells can make almost equally substantial amounts of ATP without using oxygen in a process called anaerobic respiration. In these instances, another inorganic molecule replaces oxygen. Another form of anaerobic metabolism is fermentation. Aerobic cellular respiration occurs in obligate aerobes. The process is represented by the following chemical equation. Glucose and oxygen, along with ADP, are used to produce carbon dioxide, water, and ATP. Aerobic cellular respiration can be conveniently divided into 3 stages: glycolysis, the citric acid cycle, and oxidative phosphorylation. The production of ATP by microorganisms can be achieved through glycolysis, following a cellular respiration pathway, or a fermentation pathway. Glycolysis is the first stage of energy extraction. Glycolysis is a metabolic pathway yielding ATP and NADH. The splitting of glucose occurs in the cytosol of the microorganism and involves a metabolic pathway that converts the initial 6-carbon substrate, glucose, into 2 3-carbon molecules called pyruvate. Let's look briefly at the reactions of glycolysis. The first set of glycolysis reactions are energy investment reactions. One ATP molecule is used to phosphorylate glucose, and another ATP molecule is then used to phosphorylate fructose 6-phosphate. Fructose 1,6-biphosphate is then split into two 3-carbon intermediates, glyceraldehyde 3-phosphate, or G3P. Next, we have the energy harvesting reactions of glycolysis. One electron and one proton is removed from each of the G3P molecules and used in the formation of two NADH coenzymes. G3P then becomes phosphorylated to form 1,3-bisphosphoglycerate. Substrate-level phosphorylation of ADP then occurs, forming 2 ATP and resulting in 2 molecules of 3-phosphoglycerate. 2-phosphoglycerate then loses a water molecule to become phosphoenol pyruvate. Phosphoenol pyruvate then phosphorylates another ADP molecule to form 2 more ATP molecules, leaving us with the end product of 2 pyruvate molecules. Overall, we have formed 4 ATP molecules, but at the beginning we invested 2 ATP molecules in the process. So the net gain of ATP in the process of glycolysis is 2 ATP. The next step in cellular respiration is the citric acid cycle. The citric acid cycle, also called the Krebs cycle, is a series of chemical reactions that are referred to as a cycle because the end product formed is used as a substrate to initiate the entire pathway. All of the reactions are catalyzed by enzymes and take place along the cell membrane of bacteria and archaeal cells. In contrast, eukaryotic microbes, including protozoa, algae, and fungi, this same cycle occurs in the mitochondria. There is a transition reaction that must occur with pyruvate at the end of glycolysis before it can enter the citric acid cycle. Before pyruvate molecules can enter the cycle, they must undergo an oxidation reaction. An enzyme removes a carbon atom from each of the 2 pyruvate molecules and releases the carbons as 2 carbon dioxide molecules. The remaining 2 carbon atoms of pyruvate are combined with coenzyme A or CoA to form acetyl-CoA. Equally important, the lost electrons from pyruvate, along with 2 protons, are transferred to NAD+ to form the electron carrier, NADH. The 2 remaining carbons from pyruvate are now ready to enter the citric acid cycle. Each acetyl-CoA unites with a 4-carbon oxaloacetate to form the citrate or citric acid molecule, which is a 6-carbon molecule. Let's look at the steps of the citric acid cycle or Krebs cycle. At the beginning of the Krebs cycle, acetyl-CoA loses coenzyme A and joins with oxaloacetate to form citrate or citric acid. Citrate then loses a carbon molecule and releases carbon dioxide. Electrons and protons are released to NAD+, forming NADH. Then another carbon molecule is lost to form another molecule of carbon dioxide. Another set of electrons and protons are donated to NAD+ to form NADH. Energy is then used to transform GDP into GTP. This molecule acts the same as ATP. Then more protons and electrons are donated to FAD+, forming FADH2 as another electron carrier. Then another set of electrons and protons are donated to NAD+, forming one more molecule of NADH. At this point, oxaloacetate is then reformed and can join with another molecule of acetyl-CoA to repeat the cycle. Let's look at where we are so far. In glycolysis, we gained 2 ATP molecules and 2 NADH molecules. In the transition step, we gained another 2 NADH molecules. The citric acid cycle then produced 1 more ATP molecule, 3 NADH molecules, and 1 FADH2 molecules. But that is per acetyl-CoA, and glycolysis actually yielded 2 pyruvate molecules, which both ended up being 1 molecule of acetyl-CoA. So we can double those numbers per 1 glucose molecule. NADH and FADH2 provide the starting materials for oxidative phosphorylation. Oxidative phosphorylation is the process by which most of the ATP molecules form. Oxidative phosphorylation refers to a sequence of reactions in which two events happen. The first is electron transport. The second is ATP synthesis. Here, NADH and FADH2 release their hydrogen to the carrier proteins embedded in the inner mitochondrial membrane. Hydrogen is composed of one proton and one electron. These carrier proteins separate the proton and electron of the hydrogen atom by acting as a proton pump. The carrier proteins use the electrons for energy needed to pump the protons against their concentration gradients into the space between the inner and outer mitochondrial membranes. The electrons travel from one complex to the next in the membrane and then gets picked up by oxygen to make water. This buildup of protons in the intermembrane space creates energy, which is then used by the ATP synthase to make more ATP. Protons will passively travel through the ATP synthase down their concentration gradients, which provides energy needed to turn a motor-like component of the ATP synthase. This turning allows the ATP synthase to attach a phosphate group to ADP, forming ATP. Oxidative phosphorylation takes place at the cell membrane in bacteria and archaeal cells. In contrast, in eukaryotic microorganisms, oxidative phosphorylation takes place in the inner membrane of the mitochondria. Oxidative phosphorylation is responsible for producing 34 molecules of ATP per glucose molecule. The overall sequence involves the NAD+ and FAD coenzymes that underwent reduction to form the electron carriers NADH and FADH2 during glycolysis and the citric acid cycle. In oxidative phosphorylation, these coenzymes will be reoxidized by transferring those two electrons that they had gained earlier to a series of electron carriers in the electron transport chain. These carriers are called cytochromes. These cytochromes are a set of proteins that contain iron cofactors that are able to accept and release these electron pairs. Together, the cytochrome complexes 1 through 4 form the electron transport chain. The last link in the chain is oxygen gas. Like walking along stepping stones, each electron pair is passed from one cytochrome complex to the next down the chain until the electron pair is finally transferred from complex 4 to oxygen. Oxygen also acquires two protons from the cytosol to form water. Oxygen's role is of great significance because if oxygen were not present, there would be no way for cytochromes to unload their electrons, and the entire system would soon jam up like a conveyor belt and come to a halt. Oxygen's role is also reflected in the chemical equation for aerobic cellular respiration. The actual mechanism for ATP synthesis comes from the pumping of protons through a process called chemiosmosis. As electrons pass between the cytochrome complexes, the electrons gradually lose energy. The energy, however, is not lost in the sense that it is gone forever. Instead, the energy is used at the 3 transition points to pump protons across the membrane against their electrochemical gradients, from the cytosol to the area outside of the cell membrane. Soon, a large number of protons are built up outside of the membrane, and because they cannot easily re-enter the cell, they represent a large concentration of potential energy. Protons are positively charged, so there is also a buildup of positive charges on the outside of the membrane. Suddenly, a series of channels opens and the protons flow in the direction of their electrochemical gradient. Each channel is contained with a large membrane-spanning enzyme complex called ATP synthase, which has a binding site for ATP and phosphate. As the protons rush through this channel, they release energy. This energy is captured and used to synthesize ATP molecules from ADP and phosphate. 3 molecules of ATP can be synthesized for each pair of electrons originating from NADH, and 2 molecules of ATP produced for each pair of electrons from FADH2, because the coenzyme interacts further down the chain. In a microbial cell, 38 molecules of ATP can result from the metabolism of only 1 molecule of glucose. Each NADH molecule accounts for the formation of 3 molecules of ATP, and each molecule of FADH2 accounts for 2 ATP molecules. But what about the other aspects of catabolism? Microorganisms cannot survive on glucose alone. Cells also obtain energy from other organic compounds such as fats and proteins by directing those compounds into the processes of cellular respiration. We also discover how modifications to cellular respiration and glucose metabolism allow anaerobic organisms to use glucose and generate ATP in the absence of oxygen. We find that their metabolic processes generates ATP without having oxygen gas as the final electron acceptor in the electron transport chain. Other carbohydrates besides glucose also supply chemical energy for ATP production. A wide variety of monosaccharides, disaccharides, and polysaccharides serve as useful energy sources. These non-glucose forms of carbohydrates all must go through a series of preparatory conversions before they are processed in glycolysis, the citric acid cycle, and oxidative phosphorylation. In preparation for entry into the scheme of metabolism, different carbohydrates use different pathways. Sucrose, for example, is first digested by the enzyme sucrase into its constituent molecules, glucose and fructose. The glucose molecule enters the glycolysis pathway directly, but the fructose molecule is first converted to fructose 1-phosphate. The fructose 1-phosphate molecule then undergoes further conversions and a molecular split before it can enter the scheme as DHAP. Lactose is another disaccharide which is broken in two by the enzyme lactase into glucose and galactose. Galactose then has to go through a series of changes before it is ready to enter glycolysis in the form of glucose 6-phosphate. Stored polysaccharides, such as starch and glycogen, are metabolized by enzymes that remove one glucose unit at a time and convert it to glucose 1-phosphate. An enzyme converts this compound to glucose 6-phosphate, and it then can enter the glycolysis pathway. What about protein metabolism? Proteins are broken down into individual amino acids. Enzymes then convert many amino acids to pathway components by removing the amino group and substituting a carbonyl group. This process is called deamination. For example, alanine is converted to pyruvate, and aspartic acid is converted to oxaloacetate. For certain amino acids, the process is more complex, but the result is the same. The amino acids become pathway intermediates of cellular respiration. What about fat metabolism? Fats are extremely valuable energy sources because their chemical bonds contain enormous amounts of chemical energy. Although proteins are generally not considered energy sources, cells use them for energy when carbohydrates and fats are in short supply. Both fats and proteins are broken down through glucose catabolism as well as through other pathways. Basically, the proteins and fats undergo a series of enzyme-catalyzed conversions to form components normally occurring in carbohydrate metabolism. These components then continue along the metabolic pathway as if they originated from carbohydrates. Fats consist of 3 fatty acids bonded to a glycerol molecule. In order to be useful for energy purposes, the fatty acids are separated from the glycerol by the enzyme lipase. Once this has occurred, the glycerol portion is then converted to DHAP. For the fatty acids, there is a complex series of conversions called beta oxidation, in which each long-chain fatty acid is broken by enzymes into 2 carbon units. Other enzymes then convert each unit to a molecule of acetyl-CoA, ready for the citric acid cycle. We previously noted that for each turn of the cycle, 16 molecules of ATP are derived. ATP can be produced through chemiosmosis without oxygen gas. Nearly all eukaryotic microbes, as well as multicellular animals and plants, carry out aerobic cellular respiration, using oxygen as the final acceptor in the electron transport chain. However, many bacterial and archaeal organisms exist in environments where oxygen is scarce, such as in wetland soil and water, and within the human and animal digestive tracts. In these environments, the organisms have evolved a respiratory process called anaerobic respiration that rely on terminal electron acceptors other than oxygen for ATP production. Considering the immense number of species that live in such anaerobic environments, anaerobic respiration is extremely important ecologically. The facultative species E. coli, for example, converts nitrate to nitrite. The obligate anaerobe Desulfovibrio uses sulfate for anaerobic cellular respiration. The sulfate combines with the electrons from the cytochrome chain and changes it to hydrogen sulfide. This gas gives a rotten egg smell to the environment and can be found in tightly compacted landfills. Another example is archaeal methanogens, which include Methanobacterium and Methanococcus. These are obligate anaerobes which use carbonate as a final electron acceptor and, with hydrogen nuclei as an electron acceptor, form large amounts of methane gas. Fermentation generates ATP in the absence of oxygen. In environments that are anoxic and without alternative electron acceptors, much of the organic material will be catabolized through fermentation. Fermentation is the enzymatic process for producing ATP using endogenous organic compounds as both electron donors and acceptors. Exogenous electron acceptors include oxygen, nitrate, sulfate, and carbonate. The chemical process of fermentation makes a few ATP molecules in the absence of cellular respiration. However, the citric acid cycle and oxidative phosphorylation are shut down, so the products of glycolysis, pyruvate, are shuttled through a pathway that uses other final end products. In these pathways, pyruvate is the intermediary that's accepting the electrons. In all cases, no matter what the end product, fermentation ensures a constant supply of NAD+ for glycolysis and produces 2 ATP molecules per 1 glucose molecule. For example, in the fermentation of glucose by Streptococcus lactis, the conversion of pyruvate to lactic acid is a way to reform the NAD+ coenzymes, so glycolysis can still make 2 ATP molecules for each glucose consumed. The diversity of fermentation chemistry extends to some eukaryotic microorganisms as well. For example, in yeasts, Saccharomyces, when pyruvate is converted to ethyl alcohol or ethanol, NAD+ is reformed. Fermentation is an anaerobic process that reoxidizes NADH to NAD+ by converting organic materials into fermentation end products. Fermentation end products also provide a way to identify different bacterial species. Different microorganisms have evolved a variety of metabolic pathways, generating large amounts of end products that do not directly produce ATP. Some microbial fermentation pathways have been controlled and are extremely useful to human culture and industry. Other end products are useful for the identification of bacterial species, including enteric pathogens. The energy benefits to fermentative organisms is far less than in cellular respiration. In fermentation, each glucose passing through glycolysis yields 2 ATP molecules and the final end product of fermentation. This is in sharp contrast to the 38 ATP molecules produced in cellular respiration. It is clear that cellular respiration is the better choice for energy conservation. However, under anoxic conditions, alternative. In a dairy plant, a process called lactic acid fermentation by S. lactis is carefully controlled so that the acid will curdle fresh milk to make buttermilk or yogurt. The liquor industry uses ethyl alcohol produced in alcoholic fermentation to make alcoholic beverages such as beer and wine. Swiss cheese develops its flavor from the propionic acid produced during fermentation and gets its holes from the trapped carbon dioxide gas resulting from fermentation. The chemical industries also have harnessed the power of fermentation in the production of industrial solvents such as acetane and butanol. The ability of microbes to carry out different fermentation reactions that produce different end products can be very useful in species identification. Photosynthesis. Photosynthesis can be separated into light-dependent and light-independent reactions. In the first part of photosynthesis, light energy is converted into or fixed as chemical energy in the form of ATP and NADPH. Thus, the process is referred to as the energy-fixing reactions or light-dependent reactions because light energy is required for the process. Like the reactions of cellular respiration, the energy-fixing reactions of photosynthesis are dependent upon electrons and protons. The source of these atomic particles is water. In cyanobacteria and algae, the splitting of water not only produces the needed atomic particles, it also releases oxygen as a byproduct. The ability of these ancestors of modern-day cyanobacteria to generate oxygen gas profoundly changed the atmosphere of Earth some 3.5 billion years ago, leading to the evolution of aerobic organisms that can carry out aerobic cellular respiration. Light energy is absorbed by a green pigment called chlorophyll A. Chlorophyll A is a magnesium-containing lipid-soluble compound. Chlorophylls and accessory pigments make up light-receiving formation of ATP in a process called photophosphorylation. The electrons in the photosystem I are excited by light energy and are then boosted out of the pigment molecules to the first of another set of membrane carriers, and finally to a coenzyme called nicotinamide adenine dinucleotide phosphate, or NADP+. The coenzyme functions much like NAD+, in that NADP+ receives pairs of electrons and protons from water molecules, forming an electron carrier, NADPH. In the second stage of photosynthesis, another cyclic metabolic pathway forms carbohydrates. This process is known as the carbon-fixing reactions because carbon in carbon dioxide is trapped or fixed into carbohydrates and other organic molecules. This process is also sometimes referred to as the light-independent reactions because it can occur in the absence of light. The light-independent reactions, or carbon-fixing reactions, are also referred to as the Calvin cycle, named after Melvin Calvin, who worked out the sequence of the reactions. An enzyme bonds carbon dioxide to a 5-carbon organic substance called ribulose 1,5-bisphosphate. The resulting 6-carbon molecule then splits to form 2 molecules of 3-phosphoglycerate, or 3-PG. In the next step, the products of the energy-fixing reactions, ATP and NADPH, drive the conversion of 3-PG to glyceraldehyde 3-phosphate, or G3P. 2 molecules of G3P then condense with each other to form a molecule of glucose. To finish off the cycle, most G3P molecules will then undergo a complex series of enzyme-catalyzed reactions that require ATP to reform RUBP. However, some G3P exists in the cycle and combines in pairs to form glucose. This sugar can then be used for cellular respiration, stored as glycogen, or used for other cellular purposes. Thus, the overall formula for photosynthesis can be expressed as follows: 6 carbon dioxide molecules plus 6 water molecules plus ATP in the presence of light form 1 glucose molecule, 6 oxygen molecules, and ATP along with phosphate. Notice that this reaction is actually the reverse equation for aerobic cellular respiration. The fundamental difference is that aerobic cellular respiration is a catabolic process which yields energy, while photosynthesis is an anabolic or energy trapping process. In addition to cyanobacteria, a few other groups of bacteria also can trap energy by photosynthesis. 2 such groups are the green bacteria and purple bacteria. so named because of the colors imparted by their pigments. These bacterial organisms have chlorophyll-like pigments known as bacteriochlorophylls to distinguish them from other chlorophylls. In the energy-fixing reactions, the organisms do not use water as a source of hydrogen ions and electrons. Consequently, no oxygen is liberated, and the process, therefore, is called anoxygenic photosynthesis. Instead of water, a series of inorganic or organic substances, such as hydrogen sulfide gas and fatty acids, are used as electron sources and hydrogen ion sources. Thus, the green and purple bacteria commonly live under anaerobic conditions in environments such as sulfur springs and stagnant ponds. Patterns of metabolism. Microorganisms must meet certain nutritional requirements for growth. Besides water, which is an absolute necessity, they need nutrients that can serve as energy sources and raw materials for the synthesis of cell components. These generally include proteins for structural compounds and enzymes, carbohydrates for energy, and a series of vitamins, minerals, and inorganic salts. Two different patterns exist for satisfying an organism's metabolic needs. These patterns are autotrophy and heterotrophy. Autotrophy and heterotrophy are primarily based on the source of carbon used for making cellular components. Organisms that synthesize their own foods from simple carbon sources, such as carbon dioxide, are referred to as autotrophs. Those that use light as energy sources, such as cyanobacteria, are called photoautotrophs. Microorganisms, including the cyanobacteria and algae, can carry out photosynthesis using water and producing oxygen gas in a process called oxygenic photosynthesis, while other bacterial species carry out anoxygenic photosynthesis using hydrogen sulfide rather than water, and produce sulfur granules rather than oxygen gas. Another group of autotrophs do not use light as an energy source. Instead, they use inorganic compounds and are referred to as chemoautotrophs. For example, some species of Nitrosomonas convert ammonium ions into nitrite ions under aerobic conditions, thereby obtaining ATP. The genus Nitrobacter then converts nitrite ions into nitrate ions, also as an ATP generating mechanism. In addition to providing energy to both bacterial species, these reactions have a great significance to the environment as a critical part of the nitrogen cycle. By preserving nitrogen in the soil in the form of nitrate or ammonia, it can then be used by green plants to form amino acids. Many microorganisms are heterotrophic. Such heterotrophic organisms obtain their energy and carbon in one of two ways. The photoheterotrophs use light as their energy source and preformed organic compounds such as fatty acids and alcohols as their sources of carbon. Photoheterotrophs include certain green non-sulfur and purple non-sulfur bacteria. Chemoheterotrophic microorganisms are those that feed exclusively on dead organic matter, commonly called saprobes. In contrast, chemoheterotrophs that feed on living organic matter, such as human tissue, are commonly known as parasites. Chemoheterotrophs use preformed organic compounds both for their energy sources and carbon sources. Glucose would be an example. Thank you for watching.